Okay. Okay, thank you very much, John. It's my great pleasure to be here to present our work that we're doing at EMBO and to present essentially the latest advance we have been doing with source data. As John mentioned, source data is a platform to make papers discoverable based on their data content, and also to start bridging the publishing process to the open science infrastructure, or the emerging open science landscape. So I will not have the time to go in all the facets of all the aspects of source data. I will try to present by focusing it, by presenting it as a case study, if you wish, how an AI framework can be developed to enrich the scientific content of papers, and the part we believe is really the heart of published journals. I should also say that this is in the domain of life sciences and cell and molecular biology. So our vision at EMBO Press is for the paper of the future is to have papers that provide the opportunity to users to zoom in the content and provide the information at the level that is useful for the users, from short summaries, the title and so on, but also allows the users who are interested and the expert in the field to dig into the paper, zoom in and access the data behind the paper, the computer code used for the analysis. And to this sort of vertical integration, we want also to add a horizontal integration, if you wish, by connecting the related piece of content across the literature, and especially at the level of data, that users can really explore the surroundings of a paper and find related data, related experiments, related results in a very easy way. And connecting the papers to the underlying data is very important. It is starting to bridge the publishing process with the open science infrastructure, and we have heard a lot yesterday about the importance of open science. And essentially, the data behind all the papers should be linked to the papers. This should also be extended, in my opinion, to preprint. And very importantly, the related data that are thematically related, or that test the similar experiments in different conditions, should also be cross-linked. And this combination of linking papers and data will confer more robustness to the scientific literature and also make it more discoverable. So it is to go in this direction that we have started to build the source data platform to bridge publishing to now the challenge is to see how I can try to start the movie to bridge the publishing to the open science. Can I have a mouse? Okay. So I have a trouble here. So. Let me try this. Okay. And really, we want to make the papers discoverable based on their heart, the figures. And the idea is to have enable directed search of the data that are presented in the figures, and this is really the heart of the paper. And to do this, we used the information provided by authors in the figure legends, or the labels of the figures. And we represent that in a structured way, machine-readable way, as biological entities, small molecules, drugs, proteins, genes, and so on. And we classify them whether they are the object of a perturbation or whether they are the object of the measurement. And this is a very compact representation of the experiments, and we can link them to each other to build a database, which is essentially a graph database, where experiments and figures are linked based on common elements. And we have built a series of tools now, a search interface, what we call smart figures, and also a curation interface that allows us to format the information in that form. So the search interface, the idea is that a user who is interested in a particular piece of data and experiment, and I take here a simple example where somebody would like results that test the effect of glucose on insulin. The results that are returned are papers that contain experiments that are relevant for this query, and automatically prioritize those experiments inside the paper that show the result of this experiment. Now, importantly, if we would have formulated formulated the reverse query, I'm interested now in experiment that tests the effect of insulin on glucose, the results would be different. And this is indeed a very different experiment, and this is an essential distinction for a scientist who wants to understand mechanisms of disease. And just as a little comparison, this kind of causal distinction is not made in classical search engines. The second application we have is smart figures that present data in the context of related data and experiments, and also provides a list of the key entities involved in these experiments, which are linked then to external databases. And with the smart figures, users can start navigate and hop from one paper to the other by following the links that are determined by the structure of the experiments. We realized a pilot implementation in collaboration with Wiley, smartfigures.net, where we have essentially injected the smart figure JavaScript application inside the content, and essentially the reader, by reading the HTML version of the paper, can open the smart figure within the content, and without leaving the paper, start exploring the vicinity of that paper, again based on the data links. Now, we have implemented and progressed a systematical curation process to process all the figures that we're publishing in our content. And we created a tool that optimizes that and allows us to do this in a stable way. And this allowed us to then, after a couple of months, to accumulate a fairly sizable data set of more than 20,000 experiments now across our journals, but also taking some legacy content from other journals to test the generality of our approach. And the good news is that most of the figures that we have been processing in the field of cell and molecular biology were amenable to this very simple representation, and we could capture these causal relationships and the entities. Now, of course, this is a lot of manual work. We can do that in a stable way, but it is fairly costly. So we want to reduce cost and increase the scale by automation. Now, automation of a task like this is very difficult, and it involves several steps, essentially 3 if we focus just on the text. The first one is to segment figure legends into individual panels. This seems to be trivial, but it is absolutely essential if we want to have a coherent scientific description of an experiment. Figures are too heterogeneous. There's some kind of a melting pot of all kinds of things. The second step is a classical entity recognition task for text mining, but across several entity types, different types of objects, which is very important in biology because we have to cover many scales of biological organizations, from genes and molecular level to entire organisms. But it's really the third step that is the most challenging, and we call that causality extraction. It is really to try to interpret now the meaning of these entities in the context of the experimental design and know what has been measured and what has been perturbed. Did we measure the effect of insulin on glucose, or did we measure the effect of glucose on insulin? And this distinction is really important. This is not a detail. This is actually the heart of the reason why an experiment has been done in cell and molecular biology. And before we started that project, I have to say the hopes that this would be amenable to automation was actually very, very small. And I still remember presenting this and people saying, well, no, this is impossible. So we tried to see whether it would be impossible by using a deep learning approach, and I'm not going to go now in the geeky details, but the concept is fairly simple. We use deep neural networks that can learn by the example, and we present the example of the manually curated data sets that we have accumulated. And after a couple of iterations, typically 100 or a few hundred iterations, the model is correcting itself. The neural network is pruning some links, reinforcing others, and finally is able to predict the tagging of the text in a way that is very similar to the examples that we have presented. So this is the basic of the approach, and this is the result in the form of an authoring tool, but of course we can evolve that now in in different scenarios. And since I hate online live demos that always fail, and you can call Microsoft and Apple to talk about that, I'm going to show you a movie, but the demo is live. I can show you to anybody who is interested. And here we are sort of in real time parsing the text and interpreting the text and realizing these three tasks. The first task is to segment the text into the different panels and try to be smart enough not to be misled by citations of panels within another panel. This is okay. The second step, you see the different words are being highlighted. These are the entities, the key entities of the experiments. And we now have these 7 different types of entities. You can recognize gene products, you can recognize cell lines and small molecule. But it's really the colors of these words that is the most interesting. And if you focus on the word P53, in the 2 examples, you see that once it's in blue, once it's in red, because the meaning of the same word, the same entity, it's a protein, in these different sentences is completely different. In the first sentence, P53 is the object of the measurements. This is what the scientist has observed. In the second sentence, the same word has a different context. And there, biologists will see immediately that in that experiment, P53 is the target of an intervention. The scientist has removed that gene to see what is its effect on the other ones. And it's really exciting now that we are able to have a model that is able to interpret the meaning, the scientific meaning of these entities, depending on the context of the rest of the sentence. Now, one value of machine learning and deep learning in particular is the ability to generalize beyond the examples that we have presented. And I couldn't resist to make a little example of that. I hope it's okay. Don't put it on the web. But here we have a sentence that I hope you will be convinced our machine has never seen before. This is not part of our training set, but it sort of is able to interpret in a very similar way as a human that some words look like a gene, some words look like a strange species. So you have the TRUMP1 gene. Of course, it's not a gene. Overexpression in the brain of Melania. This sounds vaguely like a Arabidopsis or something like this. And in a way, the model has identified features of those terms that make them look like a small molecule, that make them look like a species or a gene product, and essentially generalizes this and tries essentially to guess. This is also maybe a more philosophical point, whether we treat this as a success of the machine learning because the machine is sort of guessing the right thing the same as a human does, or whether we treat these as false positives. This depends on the context. If we would text mine the EMBO journal, this is perfect. If we text mine the New York Times, that would be a false positive. So here are 3 maybe lessons that I can share that we learned in that project. The first one is that machine learning, deep learning in particular, is very flexible, and there is an enormous versatility of this type of approach. And essentially here we have applied the same framework, the same architecture of deep neural networks that we have developed for that task in different contexts to do the panel segmentation, to do the entity tagging, to do the role prediction. And essentially these models are learning by the example, and it provides a quick way, or maybe not so quick, but a very flexible way to do different tasks that might be, instead of developing rigid rules for every case study. And of course, the power of generalization of these models is the true value. And the more a model can generalize, the more it actually is intelligent. And this is really where we could debate maybe in the second dinner at the bar what it means, understanding what it means intelligence. And I think there is something quite deep about the power of generalization of a computer model. The lesson 2 is that labeled training sets are extremely valuable. It is a lot of investment to gather these data sets, to curate them, to have high quality, sufficiently large volume training set. But really, if data is the new oil, the labeled data are the new oil, having this kind of data sets, labeled data sets, is opening new opportunities, and it is creating possibilities that would not have been possible before. So it is an investment, but that opens completely new doors. And finally, the third lesson I think is that we, as publishers, as editors, we tend to focus on the scientific content at the stage of publication or maybe production. And what we end up doing, like we just did, is sort of a painful reverse engineering to try to extract some elements of structure from a material that has been completely degenerated from natural language sentences that describe in a flowery way an experiment, from images that have been assembled and so on. And even if this is possible, it is difficult to scale, or it's expensive to scale. And I think we have to expand our horizon and see that the entire research workflow starts, of course, way before the time of publication. And the more we can harmonize our tools that we use in publishing, but that are also used by authors as authoring tool, and the more we can have smart tools that make the life of authors easy to preserve that structure, the more we are going to be able to scale and represent and keep the important structure of the scientific knowledge in papers. So to finish, I think AI and machine learning is a fantastic opportunity, of course, for publishers. It is a fantastic opportunity to make publishing more efficient, to make it maybe cheaper. It is a fantastic opportunity to enrich the scientific content in ways that would not have been possible before. But, and I just submit this idea, maybe it goes even beyond that, because if our goal really is to create artificial intelligence models that help us representing science, that help us advancing science, then publishers may occupy a very, very unique role, because essentially in any domains, there are journals, and these journals have access to the best experts in a routine, large-scale, scalable way, to the best experts in those fields, and these experts are the authors. And essentially, there is this natural author in the loop scheme in the publishing industry, and every publisher is in constant contact with experts. And I think this is a fantastic opportunity for publishers not only to use AI, but actually to shape it. And connecting the brain of the best experts to artificial intelligence may redefine the role of publishing and extend its mission from dissemination of papers and so on to really disseminating knowledge in a structured and computer readable way. Thank you very much.